Okay, so this lecture was just kind of an introduction to patho, as well as a review of cell biology and then some diseases associated with cellular adaptation. So we had these definitions. So patho is really just studying the changes in normal physiology that occur during disease. Pathology, this is really the study of disease at the cellular or tissue level. A disease is just a functional impairment of cells, tissues, organs, or organ systems. A syndrome is basically the clinical manifestations that you would expect with a certain disease. Okay, a specific condition with a recognizable and predictable pattern. Pathogenesis, this is the origination or development of a disease. Etiology, the specific cause of disease. Etiology is very easy when it comes to many of the infectious diseases as well as some of the genetic diseases. So etiology can be definite, like certain. In the case of an infection, a certain pathogen causes a certain disease. It can also be multifactorial, meaning there's environmental components and genetic components and a number of things that go into the etiology, or idiopathic, where the the origination of the disease is unknown, the cause of the disease is unknown. Clinical manifestations, these are the signs that accompany a particular disease, so these are things you can see. Symptoms, these are subjective, these are things the patient reports. You can't measure these, you can't see these. Clinical manifestations can be acute, meaning very rapid onset and relatively rapid to go away. They can be subacute, which is kind of on the 3 to 6 month time frame. Chronic, typically longer than 6 months. Insidious, meaning the origination of the disease is, or the clinical manifestations of the disease were kind of vague in their onset. You can't really pinpoint when they started. Okay, and then local or systemic. So obviously local, occurring in a localized area. Systemic, obviously the whole body. Morbidity basically just refers to having a disease. Okay, negative outcome with disease complication that impacts quality of life. Basically, having a disease, that's morbidity. And of course, that can result in death or mortality. Okay, then we talked about disease prevention very quickly. Primary prevention, this prevents a disease from happening. So, like wearing a helmet. Secondary prevention, this is early detection. So, like colonoscopy and other similar screening tests. And then tertiary prevention's basically rehabilitation after a disease is detected. So in the case of physical therapy and other types of therapy. All right, then we started a quick review of the cell. So of course, cells are the basic structural and functional unit of the body. All cells have three parts. They have a plasma membrane, they have a cytoplasm, and a nucleus. At least in our bodies. A couple of exceptions there, though. For example, red blood cells and platelets and things; those aren't true cells. Now, cells are able to adapt, which is good because they need to maintain homeostasis. All right. Now, adaptation, as we will see, is not effective against long-term stress on cells. Okay. And when cells are stressed for a long time, they become injured, and if the injury is too great, they will die. And there's a bunch of ways in which cells can become injured, which we'll talk about later. Okay, so we talked about some of these organelles. For example, the Golgi apparatus, that's like the UPS center, so it's going to receive things from the endoplasmic reticulum. It's going to modify them, package them, kind of ship them out. The mitochondria, these are the powerhouse of the cell. So this is where the majority of energy is going to be made. Lysosomes, these were basically the garbage cans, so they take in cellular debris and they take in products that have been endocytosed and they kind of degrade them. They can also kill the entire cell. Peroxisomes, you have a lot of these in immune cells like macrophages. They're important for killing pathogens through oxidative products, and they're important for being able to handle the oxidative products of metabolism that occurs in the cell. Rough endoplasmic reticulum produces proteins that are going to be exported out of the cell. Smooth endoplasmic reticulum, really important for the synthesis of lipids, so things like steroid hormones and things like that. The nucleus, of course, this is where the genetic material is. The nucleolus, this is where portions of ribosomes are going to be assembled, which is just this darker portion inside the nucleus. Plasma membrane, we'll talk about that in a second. The centrioles, these are going to create a portion of the cytoskeleton, as well as be important for cell division. What else here? Ribosomes, of course, these are for protein synthesis. And those are, those are kind of the big ones. Just know the basic, the gist of what these guys do. Okay, so the plasma membrane, the characteristics, it's a phospholipid bilayer. Oops. It's a phospholipid bilayer. Okay, which means it has two layers of phospholipids. So here's one leaflet, and here's the other one. They've split it here. Okay, and then within this phospholipid bilayer, you have these integrated proteins, and they do a bunch of different things. Okay, you call this is called the fluid mosaic model because it's not static. Okay, so these proteins can shift and move throughout this membrane. Okay, a couple other points here. There's a lot of cholesterol in the membrane, especially in nervous tissue and things like that. So cholesterol is very important. But also these phospholipids, they have a hydrophilic portion and they have hydrophobic portions. The hydrophobic portions face in here away from water. Water would be out here, water would be in here. Okay, so these hydrophilic, these water-loving portions face the water. Okay. Okay, so movement across the plasma membrane. So movement can be passive or active, so things can move passively or actively. Passive processes do not require energy, and that's because things are moving down a concentration gradient. Diffusion, this is the passive movement of particles, of solutes, down a concentration gradient, and there are simpler facilitated types, which we'll talk about in a second. Osmosis is the passive movement of water, okay, across a semi-permeable membrane. So some of these things that simply diffuse, oxygen, CO2, basically small lipid-soluble things. So fatty acids, steroid hormones, small alcohols, urea, water, of course, things like that can simply diffuse across the membrane. So active processes, these require energy. And that's because we are pushing things up a concentration gradient. So we're moving things from low concentration to high concentration. And it takes energy to do that. And of course the sodium-potassium pump is kind of the perfect example of an active transport pump. So here we can see passive transport. This is really simple diffusion. Things moving right through the membrane, particles moving right through the membrane itself. Alright. Facilitated diffusion would be if there was like a tunnel where things had to kind of travel through. Okay, so that's what's happening here. And then active transport, it takes energy to push things against a concentration gradient. Okay, then we talked about cell signaling, and basically cells have to communicate with each other. If they did not communicate with each other, we would be in trouble. And there are three main ways by which this happens. Okay. The first one is paracrine signaling. And in paracrine signaling, basically a cell produces signaling molecules that affect nearby cells. So you have, a cell here, and a cell here, and a cell here. And this is going to produce these molecules that affect the nearby cells. Okay, that's paracrine signaling. So a lot of things work this way. You see this a lot in the immune system. You see this with growth factors, a number of examples. Okay, autocrine signaling. Here, you have a cell that is going to produce a signaling molecule that's going to come back and cause a change in that same cell. The best examples are in the immune system, especially with T cells. But just understand what autocrine signaling is. And then finally, the one we've all heard of, endocrine signaling, is when we have the bloodstream and we have an endocrine cell that produces hormones that go into the blood. And this is going to affect various tissues that have receptors for that particular hormone. Alright, so those are the three main signaling mechanisms by which cells communicate with each other. So cell reproduction, of course, cells have to reproduce, and all of that is controlled by genes. Genes are going to control the growth of cells, the timing of their division, and their differentiation. So differentiation is when you have an immature cell, something like a stem cell, and it differentiates into its fully functional form. So a stem cell differentiates into a fat cell, an adipocyte. Stem cell differentiates into a hepatocyte, a liver cell. Okay, all that's controlled very tightly by genes. And here's the cell cycle. Don't worry too much about this. Okay, then we talked very briefly about cellular metabolism. So, cellular metabolism, the sum of all chemical reactions occurring in a cell. You have anabolic types, which are energy consuming, and catabolic types, which is energy releasing, and both are occurring. So this is all of these chemical reactions are done by the use of enzymes. All right, and of course our energy currency is ATP, or adenosine triphosphate. So basically, the production of ATP, we eat food. Okay, it gets digested into small components. Our cells prefer to use carbohydrates, specifically glucose. Okay, but they will use lipids and proteins if they need to. But carbohydrates, specifically glucose, that is the number one source of energy for our cells. So glucose will first enter a process called glycolysis, which is sugar splitting. And this process gives us 2 ATP, which is not much. Okay. Now, if oxygen's present, which in most cases it is, these glycolysis products, which are pyruvate, will go into mitochondria. All right, and then they will enter what's known as the citric acid cycle. That's where we produce CO2. And basically, the job of the citric acid cycle is to produce electron transfer molecules. Okay, and these are going to be used in the electron transport chain, which occurs across the inner mitochondrial membrane. And the big takeaway here is that this is where we produce the majority of our energy. Okay, about 34 to 36 ATP, and that's really the big takeaway. And here's just a very fun slide. All right. We started talking about cellular adaptation, which are reversible structural or functional responses both to normal and abnormal physiological conditions. And we talked about the different types, starting with atrophy. So atrophy is a decrease in cell size. Okay, this can be physiologic and normal, like enthymic involution. So basically the thymus involutes. What that means is it pretty much becomes fat as we age, or it can be pathologic. Okay, so in the case of disuse, atrophy of skeletal muscle after you break an arm or something like that. Now hypertrophy, this is an increase in cell size due to growth signals, and these are called trophic signals. This can be due to increased workload, like exercising muscles. That's a normal type of hypertrophy. Can be due to hormonal input. Okay, so hypertrophy of reproductive cells, or increased functional demand, like adenoid hypertrophy, which you would see in some infections. Hyperplasia, this is an increase in cell number. Again, this can be due to increased workload, hormonal stimulation. So you'll see this in the endometrial layer of the uterus during the monthly cycle, or pathology like BPH, okay, benign prostatic hyperplasia. And these are just showing some of these examples. So you can see we've got our normal cells up here. These are atrophied cells. You can see how they've gotten smaller. Hypertrophied cells, they've gotten larger. And hyperplasia, we've increased the number of cells. Metaplasia. So metaplasia is when one type of mature cell changes to another type. Okay? And the classic example is bronchopulmonary metaplasia, okay? Where you have these nice columnar epithelial cells, okay, and they change to these very flat, stratified squamous cells, okay? And you really don't want that, okay, because these cells have a very good function, an important function in the respiratory tract. So when they change to this, you lose that function. Okay, a similar thing happens in Barrett's esophagus in response to GERD. Sometimes you'll see metaplasia in cancer, but it does not indicate cancer. Now, dysplasia is just very unorganized cell shape, organization, size, all of that. Very unorganized growth, irregular shapes of cells, different sizes. They're in different phases of the cell cycle, and this is due to chronic and persistent stress. This typically occurs in epithelial cells. So metaplasia, so I talked about this, this metaplasia in the respiratory tract. If this continues, this can turn into bronchopulmonary dysplasia, and that's really where you start to get concerned, with cancer and things, because it can precede cancer, but it doesn't indicate cancer. So if somebody has dysplasia, it doesn't mean they have cancer. So if somebody has cervical dysplasia, it doesn't mean they have cervical cancer. Okay, so this is just showing normal ciliated epithelium. Here we have metaplasia, we change from one cell type to another, and then dysplasia, just very unorganized growth. Cell shapes are all irregular. These cells are in different stages of the cell cycle, so not good. This is just showing Barrett's esophagus. And again, just a recap of all these different types of cellular adaptations. Okay, so basically, if you have a normal cell under stress, it adapts. If it can't adapt enough, you have injury. And if that injury is mild or temporary, it's reversible, and you can become a normal cell again. But if that cell injury is persistent, it becomes irreversible, and once that injury is irreversible, you have cell death. Alright, then we talked about the diseases starting with cerebral atrophy. So this is atrophy of the neurons of the cerebrum, okay, so a reduction in size. It can be caused by injury, it can be caused by infection, it can be caused by things like stroke, which would be an ischemic cause, it can be caused by understimulation, dementia. All of those are possible causes of cerebral atrophy. What happens is, when these neurons start to atrophy, they pull away from each other. Okay, so you have different relationships between the neurons, alright? And that's because the distance between them increases, so the communication between those neurons basically stops, and that's not good. So clinical manifestations, it depends on the area, and it can be local, it can be global. So global types of atrophy you might see in certain types of dementia. Local types you might see with, strokes. And again, don't worry too much about the areas and what it might cause. Just know that it depends on where the atrophy is occurring. That's going to dictate the clinical manifestations. Okay, so for example, atrophy of the hippocampus, that's important for consolidating short-term memory to long-term memory. You see that in Alzheimer's. All right. So as far as diagnosis and treatment, diagnosis, you're trying to determine the underlying cause, and treatment, you're trying to prevent any worsening atrophy. So here's a normal brain. Okay, we talked about all of this in class, but we'll just recap. So the spaces between, or these folds here, are called gyri, and the spaces between them are called sulci. All right. Now in atrophy, what you see is you're going to see increased space, okay, between the gyri. Okay, so these sulci are getting bigger. Okay, then we talked about cardiac hypertrophy, also known as hypertrophic cardiomyopathy, and this is an increase in size of cardiac muscle cells, cardiac myocytes, due to excessive workload on the heart. Okay, so the heart has three layers. The middle layer of the heart is called the myocardium, and this is where the muscle is. This is the big, thick portion of the heart. Okay, as far as the cause, many are caused by genetic disease. They're inherited. Okay, primary cardiac hypertrophy. Now, secondary cardiac hypertrophy, these are things that increase workload on the heart. So hypertension, valve problems. I talked about aortic stenosis and things like that. That increases workload on the heart. Now, as the hypertrophy worsens, so as the increase in cell size is worse, the heart loses its compliance, its ability to stretch and contract normally. So clinical manifestations, it's based on the severity. I mentioned in class that in a lot of cases, your first manifestation is sudden cardiac death, and that's pretty bad. But besides that, things like chest pain, syncope, which is fainting, changes in heart rhythm, dyspnea, and those changes in heart rhythm, as those muscle cells get larger, it interrupts that natural conduction system in the heart. Okay, so diagnosis and treatment, genetic testing, is the big one for inherited types. Echocardiogram will kind of give you the state of the heart itself and the heart valves and blood flow through the heart. Stress tests. As far as treatment, trying to treat any underlying causes. So if somebody's hypertensive, treating that, and of course decreasing the workload on the heart. So drugs, angiotensin 2 receptor blockers, ARBs, calcium channel blockers, ACE inhibitors, all important. And we talked about what those drugs do, how they work in class. Don't worry too much about that right now, though, for this exam. So on the left here, we have cardiac hypertrophy, so you can see that the myocardium is very, very thick. Okay. And this is just BPA. This isn't hypertrophy. This is hyperplasia. All right, we talked about acromegaly, which is hyperplasia of bones due to basically too much growth hormone. Okay, growth hormone is coming from the anterior pituitary, and that's under control of the hypothalamus through a hormone known as growth hormone releasing hormone. But anyway, so too much growth hormone causes an increase in insulin-like growth factor, specifically insulin-like growth factor 1 in the case of acromegaly, and this causes hyperplasia in bones. The thing is, the epiphyseal plates or the growth plates have closed, so this occurs in adults. All right. If this is occurring in children, too much growth hormone is occurring in children, then that's called pituitary gigantism. Okay, but this is in adults. So bones do not get bigger in length or longer. They get bigger in width and just overall mass. Okay. So yeah, this is a disease of hyperplasia. So clinical manifestations, the big ones, enlarged hands and feet, and changes in facial structure. Okay, so you'll see very prominent brows and changes in the shape and size of the nasal bone and the jaws. Okay, but there are some other clinical manifestations like soft tissue swelling. You'll also see that in the face, and pain and numbness in extremities. Okay. So diagnosis based on too much IGF-1 as well as growth hormone, and treatment, you're trying to decrease growth hormone production. So medications, but also a lot of times surgery, because this is often due to a tumor of the pituitary gland. So here you can see kind of a progression of this gentleman here. So you can see as we kind of go along, you can see changes in the brow here, and then you really start to, you see a little bit in the nose here, but you really start to see it in the nose here, and then obviously this is kind of the end product of acromegaly. Alright, we talked about cell death. So like I mentioned earlier, when cell injury is irreversible, cells die. And there are a couple ways by which they can do this. One is apoptosis. So apoptosis is basically programmed cell death, okay, or cellular suicide. And this is going to happen generally on a smaller scale than necrosis, which I'll talk about next. And this is a normal process. It happens every day. It's happening right now in all of us. But sometimes it's associated with pathology. So, in the case of like an infection, we talked about interferon with viral diseases. And interferon can signal other cells to basically commit cellular suicide. So that's just an example. But also other stressors can cause cells to undergo apoptosis. Okay, necrosis. This is death of cells related to cell injury, typically on a larger scale than apoptosis. And necrosis is never normal. And there are a number of ways by which this happens. Ischemia is a really common one for necrosis to occur. And you can see some examples down here of how lack of blood flow causes cellular death and eventual necrosis. And this is just kind of showing differences between apoptosis and necrosis from a normal cell. Don't worry too much about those details. So cell injury mechanisms, so the ways in which cells are injured, there's a number of ways. Mechanical injury, so this is direct impact, okay, like a scrape of the skin or, a crushing blow to, the femur or something. Thermal injury, temperature extremes, so burns and frostbite. Toxic injury, so, chemicals or toxins that might be coming from our own body or, exogenously from somewhere else. And then deficit injury, this is when cells are deprived of things. So deprived of oxygen, which is specifically called hypoxic injury, deprived of water, lack of hydration, deprived of nutrition, so on and so forth. Now ischemia, most common cause of hypoxic injury. And ischemia is different than hypoxia. Hypoxia is a lack of oxygen in the cells, in the tissues. Ischemia is basically the lack of oxygen due to a lack of blood flow. And this can occur through, vessel blockage, through a narrowing of arteries, through, ruptured aneurysms, all sorts of things. And then we finished talking about all of this with oxidative stress. So oxidative stress, this is when reactive oxygen species, or ROS, overwhelm the antioxidant systems of the body. Our bodies produce free radicals, which are basically atoms or molecules that have an unpaired valence electron, which is very unstable. And these free radicals can basically affect any part of the cell. But they're normal. They form normally all the time. But we have ways to combat them. We have antioxidants, okay, like vitamin E and vitamin C, and enzymes like superoxide dismutase and a number of other ones. Don't worry about any of those. Just know that free radicals are normal, and we generally have ways to fight them. However, they do occur during diseases. Okay, inflammation's a big one, and sometimes those free radicals can overwhelm our systems. And when it really gets bad is when they start to affect nucleic acids, and they start to oxidize nucleic acids like DNA and RNA, because that can lead to mutations, and mutations leads to possible dysregulated cell growth, and that of course can lead to cancer. This is just showing mitochondria. So mitochondria is a hotbed for all of these radicals. Don't worry too much about that. All right, then infectious injury. So obviously pathogens are pathogenic, and they cause injury either by production of toxins, through hypersensitivities, through invasion of cells or destruction of cells, a number of ways by which they injure our own cells. Now, one new topic I introduced was immunopathology. And this is when the immune response to a pathogen is damaging to our own cells. So it's not so much the pathogen, it's our immune response to the pathogen. That's why there are so many diseases that have flu-like symptoms, because those symptoms aren't specific to a pathogen, they're specific to the immune response. So the immune response is, trying to fight these pathogens, and that's why you get these symptoms. Like the common cold or the flu, those symptoms really aren't due to the virus. They're due to our body's response to the virus. So that's called immunopathology. Okay? As always, if you have any questions, please let me know.